Hello, my name is Heather Torrey, and I'm a research scientist at IMV, a clinical stage biopharmaceutical company pioneering a new class of immunotherapies and vaccines based on our novel platform DPX. Today I will present DPX as a platform for lymph node targeted delivery of drugs and immunomodulatory agents. Generally, these agents are systemically administered. However, many are active primarily on specific immune cells, such as dendritic cells or T cells. Since the lymph node is the primary site for priming and activation of immune cells, the purpose of this study was to evaluate the efficacy of DPX for targeted delivery to the lymph node. Our approach, as shown in Figure 1, was the formulation in DPX of 3 types of agents, selected specifically for evaluation in our murine model of 3 drug delivery parameters: distribution, targeted effect, and functionality. First, DPX formulation of small molecule dyes enabled us to visualize the distribution and kinetics of lymph node-specific delivery. We show in Figure 2 that with DPX dye formulations, there is clear sequestrations of the dye in the subcutaneous depot, or within the regional lymph node, and this is in contrast with aqueous dye formulations, which lead to widespread dissemination of the dye. Analysis of serum samples, shown in Figure 3, further demonstrates that with DPX, there is minimal to no systemic exposure. To evaluate distribution in specific cell types of various tissues, we used flow cytometry. In Figure 4, we show that with DPX dye, there is gradual uptake of the dyes in the dendritic cells and T cells of the lymph node, whereas with aqueous dye, there is quick uptake and widespread distribution of the dyes in all tissues and all cell types. As expected, there was no dye detected in our controls. Next, to evaluate lymph node targeted effect, we formulated a small immunomodulatory agent in DPX and evaluated lymph node specific impact. Cyclophosphamide, or CPA, is an agent that, when delivered systemically, leads to immune cell depletion in vaccine-draining lymph nodes. In Figure 5, we show that DPX delivery of CPA resulted in a significant reduction in the number of lymph node cells after a single administration, similar to the systemic treatment, which received 7 times as much CPA. This demonstrates that with DPX, equivalent lymph node-specific activity was achieved with significantly less active agent. Finally, with the checkpoint inhibitor anti-CTLA-4, we demonstrate that biological functionality of large macromolecules is maintained in the DPX formulation. Using the C3 tumor model and our vaccine DPX-FP along with metronomic CPA, we show in Figure 6 and 7 that mice treated with DPX-FP plus anti-CTLA-4 exhibited improved survival rates and reduction in tumor growth that was equivalent to systemic administration of anti-CTLA-4. Furthermore, anti-CTLA-4 delivered by DPX binds to CD3 positive T cells even in the presence of anti-drug antibodies. With analytical testing, we shown in Figures 8 and 9 that there is no degradation of CPA or anti-CTLA-4 in the DPX formulation. To summarize, we first show with dyes that DPX can deliver small molecules to the lymph node with minimal to no systemic exposure. Second, with CPA, that using DPX can provide lymph node targeted delivery with significantly less active agent. And finally, with anti-CTLA-4, that the functionality of large biological macromolecules is maintained in DPX formulations. Our proposed mechanism of action is shown in Figure 10, where DPX formulations are injected subcutaneously and carried from the site of injection by dendritic cells to the regional lymph node, where they interact directly with immune cells. This approach may improve drug delivery over systemic administration, resulting in increased efficacy, the use of less agent, and reduced off-target toxicity. While many other approaches to targeted therapies have failed to provide therapeutic index commensurate with their promise, the main challenge has been achieving selectivity for the target cell type. Here we show that DPX delivery platform has the potential to overcome this challenge, thereby increase the efficacy of immune and cancer therapies. I would like to thank everyone involved in collecting and analyzing this data at IMV, and thank you all for your attention. For further information, please contact Dr. Marianne Stanford at mstanford@imv-inc.com. Thank you.